In this video, I will show you how to complete a Punnett square analysis chart for use with Mendelian genetics problems. We will start with some ground rules. When we are studying a single genetic trait, only one letter will go in the outside boxes and two letters in the inside ones. We will copy the letters from top to bottom and left to right. Staying organized is very important. I have highlighted with colors to help you see how the chart is completed and how we interpret the results. In the first example, we will be using two parents who have straight hair and heterozygous genotypes. Parent 1, typically the father, goes on the left side of the diagram. In this case, the parent has the genotype big T, little t. Let's fill one gene into each box. Parent 2, typically the mother, goes on top of the Punnett square. We then fill the chart as shown. As you saw before, both parents had heterozygous genotypes and straight hair phenotypes. What does the Punnett square tell us about their possible children? Here we can see that 1 out of the 4 children, or 25%, will have the big T genotype shown in purple. 2 out of 4 children, or 50%, will have the big T, little t genotype shown in blue. And 1 out of 4 children, or 25%, will have the little t genotype shown in pink. We write in a ratio of 1 to 2 to 1, using the genetic codes for clarity. The phenotypic ratio tells us the likelihood that any child would have curly or straight hair. The bright blue boxes highlight all genotypes with a dominant T. 3 out of 4 children would have the dominant straight hair texture, a 75% chance. In red, we can see that only 1 in 4 children will have only recessive traits, a 25% chance, and they will have curly hair. Next, we will consider dihybrid crosses. These are crosses in which we would like to know the likelihood of 2 different genes occurring at the same time. We assume that each of these genes moves independently, although we will see that in some cases this does not happen. The first step, when we are looking at 2 different traits, is to figure out all of the possible genotype combinations that could be formed in the sperm or eggs. In this example, parent 1 has a heterozygous, or big T, little t for the hair texture gene having straight hair, and a heterozygous genotype for the hairline trait big W and little w, giving a pointed hairline at the top of the forehead called a widow's peak. Because each of these gametes has the opportunity to line up on either side of the meiosis I metaphase plate, the genes can appear in any order, so long as each egg or sperm has a gene for both traits, hairline and texture. In this case, the father has 4 different possible combinations of traits in his sperm: big T, big W, big T, little w, little t, big W, little t, and little w. Because we are talking about a haploid sperm, each one will only have one version, or allele, of each trait. Here we have the 4 possible sperm from the father and 4 possible egg combinations from the mother. I have used individuals heterozygous for both traits for this particular cross. 2 different letters representing the different traits go in the outside boxes. The traits are filled across from the top to bottom, just like the smaller monohybrid cross Punnett squares. First, I will populate the Punnett square with the T's representing hair texture, and then I will populate it with the W's representing hairline. I will methodically fill in the chart, keeping T's together and W's together to make it easy to read. Each inner box should end up with 4 letters representing the father's contributions for texture and hairline, and then representing the mother's contributions for hair texture and hairline as well. When we analyze this larger Punnett square chart, we have 4 possible combinations for each parent's genes for each trait, and 16 possible opportunities for them to interact. We can look at the Punnett square that there are 4 different genotypes that would produce the phenotype in which both dominant alleles are present for both traits, shown in blue. This gives a 9 out of 16 chance that the child will have straight hair with a widow's peak. There are 2 different possible options in which there is a dominant allele for straight hair texture, but a recessive allele for hairlines yielding no widow's peak. You can see that these are shown in purple, leading to a 3 in 16 chance of the straight, no peak phenotype. There are also 2 possible options in which there is a recessive allele for curly hair texture, but a dominant allele for hairlines, yielding a widow's peak. You can see that these cases are shown in pink, leading to a 3 in 16 chance of the curly widow's peak phenotype. Lastly, since both parents do have recessive genes for both traits as part of their genetic complement, there is a possibility that a child will inherit only recessive traits, leading to a 1 in 16 chance of having curly hair with no widow's peak. The genotype that corresponds with a fully recessive phenotype is shown in yellow. I hope this video has helped to clarify the process by which we set up and analyze Punnett squares. After you have watched it a few times on your own, try some of your genetics practice problems. I highly recommend that you use colored pencils to help organize or color code your own squares to better help you visualize and keep track of the information. Soon you will find that doing Punnett squares is no longer harder than filling out any other type of chart.